La 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 la, I'm a happy little girl. Eee, what a cute squirrel! Come here, little squirrel. You're so cute. Yes, you are. Grrr. Ow, stupid squirrel! Get off of me! I'm gonna go get my mommy. I don't feel so good. The zombie virus spread until it reached the university on the outskirts of town.
Now that we've gone over the syllabus, let's start with the basics of organic chemistry. Does everyone remember hydrogen bonding from general chemistry? Obviously we all remember that. Do you think we're stupid?
Could you go over it, Professor? I could use a refresher.
Of course. Alright, hydrogen bonding. Elements have different electronegativities, or affinities for electrons. This means that some atom species will pull electrons toward themselves more than other species will. Within a molecule, the more electronegative species will pull more of the electron charge towards itself. This creates a slight difference in polarity between atoms. Polarity refers to the separation of charges, in this case meaning that one atom is slightly positive and the other is slightly negative. The negative atom is the more electronegative one, since it pulls the negatively charged electrons towards itself. This leaves a slight electron deficiency in the other atom, causing a slight positive charge. A partial negative charge of an atom in one molecule can form a temporary association with the positively charged atom from another molecule of the same type, or another polar molecule or atom. This is an intermolecular force called a dipole-dipole interaction. In the case between hydrogen and oxygen, a special type of dipole-dipole interaction called hydrogen bonding occurs. Hydrogen bonding typically occurs between hydrogen and either oxygen, nitrogen, or fluorine, due to the extreme differences in electronegativity. Bro, what does hydrogen bonding have to do with, Chem? I thought that stuff had to do with, water. Is there, nitrogen in water?
Oh my god. How are you in this class right now? Seriously? Nitrogen in water? It's H2O.
How did you even get into this school? UC Lysine is known for its vision in biological research. OChem is the foundation for biological research, and you don't even know about hydrogen bonding?
It's okay, Jack. I didn't know either.
Class, let's settle down. No more rude comments. Everyone learns at their own pace. Plus, you've had a whole summer to forget everything. No, Jack, water is not the only thing that undergoes hydrogen bonding, but it's a great example. In organic chemistry, important molecules that undergo hydrogen bonding are amino acids. Amino acids are the building blocks of proteins. They have a skeleton made up of an amine group, a carboxylic acid, and a side chain called an R group that varies amongst the different amino acids. 11 of the amino acids can undergo hydrogen bonding via the different groups in their side chains. Amino acids link up to make polypeptide chains. They do this through peptide bonds, where the amine on the skeleton of one amino acid bonds to the carboxylic acid on the skeleton of another amino acid. Those chains fold into macroprotein structures, and hydrogen bonding interactions help define how those structures form and how proteins interact with each other.
What?
Zombies! Zombies! Ah! Over here, broski.
Oh my god.
Oh my gosh, did you see that? Those were zombies! They ate my professor's face off! What symptoms did these people portray? What was wrong with them? They ate people's faces off! They were slobbering and jerking like they were trying to move but couldn't control themselves. Some looked terrified, some angry, and then they started attacking other people, biting them. Oh god, that cute cheerleader was the one who ripped the professor's face off. We are all going to die! Bros, we gotta do something, man. We gotta protect ourselves. Ah! Huh! Ah! Ah! Ugh! I have to be a hero. I'll help hold the door. We can't hold the door much longer. Boom. I'll save you. No!
Oh my God!
No worries, everyone. I have my Epic Scope 3000. It can tell the structure of whatever is infecting these people. One moment. First, we place this arm in the in end of the microscope. Then you wait for it to process, and out comes your data. It truly is a wonderful machine. It looks to me, based on the data, that we have a mutant strain of the rabies virus never before described. The rabies virus is bullet-shaped and has 5 different proteins. The G proteins, or glycoproteins, on the outside are used to attach to host cells and also are the sites for antibody attachment. In this particular strain, the P protein, which is a cofactor that binds to RNA polymerase, seems to be very advanced in how it attaches to dynein light chains. The light chains seem to be used in moving organelles across cells and may also have an influence in rabies transmission because of their association with the P protein of the rabies virus. 2 amino acids, glutamic acid and glutamine, are associated with the hydrogen bonding between the P protein and the light chains. The deprotonated oxygen in the glutamic acid hydrogen bonds with a hydrogen from the amine in the glutamine. This and other hydrogen bonding and intermolecular interactions causes the 2 proteins to bind together. This association between the light chains and the P protein seems to be associated with the spread of the virus to the central nervous system. In our mutant strain, this process is severely increased so that virus takeover is only a matter of seconds. With my handy-dandy chemistry set and my supply of non-lethal rabies, we may be able to whip up a cure in no time flat. To do that, we need to stimulate the immune system to make antibodies that will attach to the proteins on the outside of the rabies virus. Antibodies are site-specific, so they make a particular antibody for each individual antigen. This means that the protein structures of the antibody and the antigen are very important in order for the 2 to bind together. For example, in hepatitis B, the antigen has a threonine on it, and so does the antibody. These 2 molecules will hydrogen bond. One of the oxygens from the hydroxy group will hydrogen bond with the hydrogen from the other hydroxy group in the threonine. We will use this knowledge and the power of chemistry to create a cure for this rabies mutant. And voila! A cure! And to distribute the cure, we'll use my handy-dandy bazooka! Bam!
Yay! We're cured!
You've done it again, science.